Chapter 10, Muscle Tissue, Part 2. So, as we mentioned at the end of last video, the muscle fiber has a plasma membrane called the sarcolemma. It's filled with cytosol, but it's referred to as sarcoplasm, and it has these long protein rods going its length called the myofibrils, and they are important for the contraction of the skeletal muscle fiber. Here's just another view of a skeletal muscle fiber, the sarcolemma. There's the multiple nucleuses, many mitochondria, the long myofibrils. We also see in this picture another important structure for skeletal muscle fibers called the transverse tubule. Transverse tubule is an invagination of the sarcolemma that travels through the width of the muscle fiber. It is basically a tunnel made of the sarcolemma going through the cell, and therefore it is filled with the interstitial fluid, the same fluid surrounding the rest of the cell. And these tunnels, the transverse tubules, wrap around the myofibrils as they're traveling through the skeletal muscle fiber. And this is important because of that whole movement of charge along the length of the skeletal muscle cell. So there are many sodium-potassium ion pumps that are found all along the sarcolemma, and the sodium-potassium ion pumps create a gradient where you have a high concentration of sodium ions outside of the cell and a high concentration of potassium ions inside the cell. And the way it does this is it will send 3 sodiums out and then bring 2 potassiums in. And this, of course, requires the expenditure of ATP. And so these important transmembrane proteins maintain that distribution. High sodium ions outside, high potassium ions on the inside. And this actually causes a charge distribution across the sarcolemma, so that we have a negative charge on the inside of the cell and a positive charge on the outside of the cell. And this is referred to as the resting potential. So at rest, when the muscle is not doing anything, its charge distribution is negative 95 millivolts. Negative 95 millivolts. And this is important because this is needed to then allow us to lead to excitability and conductivity to allow for that current, that muscle action potential to be generated. All right, so when a muscle action potential is generated, we have these voltage-gated ion channels. So there's a change in charge over where the axon stimulated the sarcolemma via the binding of neurotransmitters. And this change in charge leads to the opening of the voltage-gated sodium ion channels. When they open, sodium ion floods into the cell, going from high concentration on the outside to low concentration on the inside. And this is what leads to the muscle action potential that will then travel the length of the cell and also travel down into the transverse tubules, going deeper into the cell. And then slightly after that, the voltage-gated potassium ion channels will open. Potassium ions will flood from inside to outside of the cell, and this will end the muscle action potential and help to reestablish the original charge across the sarcolemma. The important thing is this charge, this muscle action potential, will then go down the transverse tubules. So it is the opening and closing of ion channels, voltage-gated ion channels, that cause the electrical change, that cause the muscle action potential, and sends it down the transverse tubules. So here, for instance, is a transverse tubule, say. The resting potential was disrupted because neurotransmitters bound to the skeletal muscle fiber, changing the charge in that area. That then causes the voltage-gated sodium ion channels to open. Sodium ions flood into the cell. This then changes the charge at that place. That's the muscle action potential. This change in charge then stimulates neighboring sarcolemma to open its voltage-gated sodium ion channels. And so they open, sodium ions flood in. Of course, what happens over here after that is the voltage-gated potassium ion channels open, reestablishing the original charge distribution. However, here we now have the muscle action potential. It is now in this area. It will then stimulate the next neighboring area to have its membrane voltage-gated channels to open, and that will cause the charge to travel down the transverse tubule, down along the sarcolemma. So, original change in charge leads to a movement in charge by causing voltage-gated ion channels to open and close along the length of the sarcolemma and also down into the transverse tubules. All right, next up is the sarcoplasmic reticulum. It is another specialized structure found in skeletal muscle fibers. It is similar to smooth endoplasmic reticulum, except it specifically wraps around the myofibrils, and its main job at rest is to store calcium ions. So the sarcoplasmic reticulum stores calcium ions when the muscle fiber is at rest. And as you can see, the way it is arranged is it goes between adjacent transverse tubules. So you get a structure referred to as a triad, where you have the terminal ends, the terminal cisternae of two sarcoplasmic reticulum next to a transverse tubule. So that is the triad. And this is important arrangement because when that muscle action potential travels down the transverse tubule, it will trigger the sarcoplasmic reticulum to release its calcium ions. So once that muscle action potential makes it down into the transverse tubule, it will trigger the sarcoplasmic reticulum to release its calcium ions. And the release of calcium ions is what triggers the muscle contraction. So releasing calcium ions from the sarcoplasmic reticulum causes the muscle to engage in contraction. So here we have the membrane of the sarcoplasmic reticulum, lots of calcium ions inside of it, some calcium ions outside, and calcium ions are constantly being pumped into the sarcoplasmic reticulum and then bound to proteins to help in the storage of the calcium ions. So that is when it is at rest. Energy is being expended to pump calcium ions into the sarcoplasmic reticulum. Here is the transverse tubule. When the muscle action potential is in the transverse tubule, traveling down the transverse tubule, it will, of course, be relatively close to the membrane of the sarcoplasmic reticulum, the whole triad setup. This change in charge then affects the sarcoplasmic reticulum's membrane by activating voltage-gated calcium ion channels. And then calcium ions will flood out of the sarcoplasmic reticulum through those voltage-gated calcium ion channels, going from high concentration to low, and thereby initiating the muscle contraction. So the change in charge, the muscle action potential, causing sodium ions to flood into the cell through the walls of the transverse tubule, will trigger voltage-gated calcium ion channels in the membrane of the sarcoplasmic reticulum to open, and then calcium ions will flood out, initiating the muscle contraction. And this brings us to the myofibrils. The myofibrils, referred to as the contractile element of the cell, it is the myofibrils, these dense protein structures that get shorter, cause the contraction, cause the muscle skeletal fiber to get shorter. So if we take a close-up look of the myofibril, we see that it has a reoccurring structure called the sarcomere. So here is the ends of the sarcomere as a whole. Sarcomere is composed of the myofilaments, those protein rods. And the sarcomeres are attached to each other end by end so that you end up with thousands and thousands of sarcomeres making up a single myofibril. And so it's the sarcomeres in the myofibril that get shorter, that cause the myofibril to get shorter, that cause the muscle cell to get shorter. There are two types of myofilaments. There are thick filaments and thin filaments. And the thick filaments are thicker than the thin filaments. There are also some specific structures within the sarcomere that have been given names. The dense protein structures at the ends of the sarcomere attaching it to its neighboring sarcomere, those are called Z-discs. So Z-discs are at the ends of the sarcomere. Then down the midline, the middle of the sarcomere, is a structure called the M-line. So another protein structure called the M-line running down the middle of the sarcomere. Then we have regions. So the A band is the region of the sarcomere where the thick filament is present. So the length of the thick filament is the length of the A band. Then we have the H zone. The H zone is where you only find thick filament. There is no thin filament here, only thick filament. So that is the H zone, only thick filament. And then the I band is at the ends of the sarcomere, and it is where there is only thin filament and no thick filament. So I band, only thin filament, no thick filament. H zone, only thick filament, no thin filament. I band, length of the thick filament. And of course, there are zones of overlap where we have both thick and thin filament. Here is a view of a sarcomere using electron microscope. So here's the Z discs, here's the M line, the H zone where there's only thick filament, I band where there's only thin filament, and of course places where there is overlap between thick and thin filament. And these reoccurring dark and light regions make up the striations of the skeletal muscle tissue. So again, dark and light banding represents regions of the sarcomeres. And since they're all lined up along the length of the skeletal muscle cell, we get dark and light, dark and light, dark and light, all the way along the length. Pretty impressive that we can see these protein structures with our microscopes. Thick filament. Thick filament is made up of myosin. Lots and lots of myosin proteins make up the thick filament. The myosin proteins are contractile proteins, so they actively shorten the sarcomere. And they do this by initially binding to actin proteins in the thin filaments. So myosin proteins make up the thick filament, and myosin is a contractile protein that binds to the actin proteins in the thin filaments and actively cause the sarcomere to get shorter. Then we have the thin filaments. Thin filaments are actually made up of 3 different proteins. So we have actin protein. So these yellow spheres represent actin. And actin is the other contractile protein. Actin, surprise, surprise, has myosin binding sites. So myosin can bind to actin. And so actin and myosin together lead to the shortening of the sarcomere. Then we have tropomyosin. Tropomyosin are represented by these long brown rods. Tropomyosin is a regulatory protein, and it covers the myosin binding sites on actin. So it acts as a toll gate, preventing the myosin from binding to the actin. And then these blue structures represent the troponin. Troponin is another regulatory protein. And troponin is attached to tropomyosin, and when troponin binds to calcium ions, it changes shape, and when it changes shape, it moves the tropomyosin, causing the binding sites, the myosin binding sites, to be exposed on the actin. So the troponin is what causes the toll gate to rise up to allow the vehicle to go through. So again, tropomyosin covers the binding sites for myosin, and troponin, when bound to calcium ions, changes shape, moves the tropomyosin, thereby revealing the myosin binding sites on actin by allowing the contraction to occur. Regulatory proteins. And that is where we will stop for this part of the lecture.